my great pleasure to introduce Barrett Katz, who is the Chief Medical Officer at GenSight Biologics, and his talk will be entitled, What We Have Done, What We Are Doing. And Barrett, let me tee you up here. There you go.
Delighted to be here. If I had been forewarned that I would follow Nancy, I'm not so sure I would have accepted. But it is what it is. Now listen, I'm going to give you a helicopter view. I want you to see what we've seen as a company. I want to share with you what we've been doing, and I want to share with you the results we have so far. And I want to preface my remarks by saying this is a marathon for all of us. This is not a sprint. And so let us remember we're at the beginning of this journey, nowhere near the end. We as a company address ultra-orphan diseases. Mitochondrial diseases, frankly, is first and foremost on our list. We've done 2 clinical trials for Leber disease, and the difference in the clinical trials are that in one, the visual loss was from 0 to 6 months for patients to be enrolled, and for the second, the protocol would be the same, except that the visual loss was from 6 to 12 months. As these studies were put together several years ago, of course, we've learned more and the world has changed. And so what we thought then is a little different than what we think now. And of course, now we're wondering, as Nancy said, when is the best time to capture successful intervention for the Leber patient? I want you to look at the chart on the right here. That is a high contrast ETDRS chart. It's the chart that you will see in your ophthalmologist's office when he or she turns down the lights and turns on the projector. It's very black letters against a very white background. It's what we use, generally speaking, to measure central acuity. But I add parenthetically, the world is not a black and white world. The world is much more like what we see in this room. There are areas and differences of grays. And so we're going to talk about this as we move on. In our subjects in this first clinical trial, what was called REVERSE, we had 37 subjects. They were young folks, as you would think, mostly male. Their visual acuities were assigned as they came in. The patients were randomized so that some were put in a treatment group to get drug in one eye, and in fact, a sham injection in the other eye. And what we found was that they were pretty similar coming in the one eye to the other. This drug that was used, it's a gene therapy drug with an adenovirus type 2 vector, very safe, remarkably safe, injected in the eye. And I would say to you that when you first hear about injections in the eye, you cringe, but I will tell you they're done with a needle that is so fine, most patients rarely complain of anything after such intervention. There were some side effects to these therapies, a little inflammation in the front of the eye and the back of the eye. When we sterilize the front of the eye to do the injection, what we use to sterilize the eye causes some inflammation of the cornea. But short story, this intervention, this gene therapy, remarkably safe. Okay, now we're going to talk about the results from this first trial that we looked at. And again, I want you to get the helicopter view. No one bit of information is overly compelling or convincing or valuable here. It's the totality of the picture that helps us and lets us decide where to go next. What we saw in this trial was that the temporal quadrant of the nerve fiber layer in the eye, where the papillomacular bundle lives, where the most important axons exist that subserve central fovea vision, there seemed to be protection of these nerve fibers from this drug, comparing the sham injection to the drug injection. Now, what's a sham injection? It's an injection in which the eye is prepared as though it were getting a real injection with topical anesthesia, a little bit of an injection in the subconj area, the outer part of the eye, speculum is put in, but a syringe without a needle is pressed against the eye. So that's the sham injection. That was our control in this study, for better or worse. Second observation to be made in this study, the retinal ganglion cells, which are those cells that are in fact most affected by Leber disease, the ganglion cell volume in the drug-treated eye was much more preserved than in the sham-treated eye. And so you might say, well, those two bits of information are very, very nice. They tell us that for bioanatomic markers, there may be an efficacy signal here from this novel gene therapy. And so that was what we saw in this study. Bioanatomic markers do not correlate with vision. You can have good vision, bad anatomy, bad vision, good anatomy, any mix you want. But nonetheless, when you see a change in these bioanatomic markers, remember that the OCT, which is a quick scan of the eye, is objectively measured, which means that the patient doesn't say, yes, I see this letter, or no, I don't see this letter. It's like getting an X-ray. The patient sits there, the procedure is done, the results are kicked out by the machine. Now let's look at something else in this study, visual improvement. Strangely enough, atypically, unexpectedly, both eyes, the sham-treated eye and the drug-treated eye in this trial improved. 0.2 logMAR units, that's about 10, 11 ETDRS letters. 2 lines of vision for Snellen acuity equivalent. Let me say it again. The drug-treated eye improved. Surprisingly, the sham-treated eye, which did not get drug, also improved. Now, these were very unexpected results. Is this signal? Is this noise? It certainly is counter to what we thought we knew about the natural history of this disease. In other words, these patients are followed for a year after their visual loss. We don't expect them to get better. And yet, our patients who had no drug treatment in the no drug eye got better. Could this be placebo effect? Could it be noise? We are not yet sure what this means, but we are forced to question our assumptions and say to ourselves, maybe there is a bilateral effect of this drug injected in one eye on the other eye. So that's just one of the possibilities, which takes a while to even begin to think about, because it's counterintuitive, and much of the world is counterintuitive. What else can we look at and make sense of what we saw in terms of acuities? Well, once you see this result, you've got to say, geez, what the hell does this mean? So you've got to go to the literature, talk to the clinicians, and talk to neuroscientists, other than the doctors who take care of these patients. And the neuroscientists say, gee, we come across this in our work. We do see effects in the untreated eye. And the scientists who do research in other optic neuropathies can tell you, well, I've got this experimental model. I treat one eye and I see something in the other eye. So maybe there's a possibility of a bilateral effect. We have seen it in the anti-VEGF world for macular edema, and it is something that the retina guys will talk about. What other results did we see from this reverse trial? In terms of visual fields, no significant difference between the drug-treated eye and the sham-treated eye. However, here's the showstopper, contrast sensitivity. Well, what the hell is contrast sensitivity? Contrast sensitivity, many of you in the audience, myself included, will remember the old black and white TVs. On those old black and white TVs, if you turn down one of these knobs, if you turned it down, it got more and more gray to the point where you couldn't really discern anything in the picture. Look at this chart here. The top line is very, very black against a very white background. The line down is a little more gray. The line down below that is more gray still. The line below that is more gray still. And this is what contrast sensitivity looks for. In other words, it's very easy to recognize the V, R, S, K, D, R letters. It becomes more difficult to recognize the C, N, H, Z, O, K letters. And in fact, there are 3 lines below that one. But what we're measuring on this chart is the ability to discern low contrast changes of resolution. And frankly, for our population of labor patients, this is a much more meaningful real-world metric than going to your eye doctor, sitting in a darkened room, having that projector turned on, and looking at very black letters. What we saw in our study, this first phase 3, is that the contrast sensitivity was almost doubled in the eyes that had drug treatment. And that's a very encouraging sign. It is a recognized visual function. It's not a visual function most people are familiar with. Nonetheless, it's very real. We have other data to show you that there are different ways to look at this data and cut and slice it and say, wait a minute, these patients had a drug treatment, they gained 6 letters more than the sham eye. In some circumstances, if we look at the best treated eye versus the best sham eye, they gained 12 letters as opposed to 4 letters. There's all kinds of positive information in that study from which we can learn. In fact, one of the things we saw in the study was if the endpoint of that study had been eyes that reached the level of 2200 or better, the study would have met such a primary efficacy endpoint. Let me give you some summary statements here. In a study like this, in any clinical trial, we have a binary event. You choose a primary efficacy endpoint, you meet it, you get it, or you don't. But as we talked about, the world is not a black and white world, it's a gray world. And so you've got to dig into these studies to see what information might be there. We found in this study that drug-treated eyes were more likely to achieve a vision of 2200. We found statistically significant protection of the retinal ganglion cell and the temporal quadrant of the nerve fiber layer. We found that the contrast sensitivity of the drug-treated eye just about doubled. Again, what else did we see? For subjects with better acuity at entry, they did better. When the visual loss was for a shorter period of time, the subjects were more likely to do better. If we define responder rates differently, we can see a real signal here. But again, that was a reverse study. The second phase 3 trial, what we call Rescue, will be out in fourth quarter of this year, December, perhaps January. We're starting another trial called Reflect, in which there'll be bilateral treatment, and we're installing a control group in that study. I want to close with this comment. We're all in this together. Do not be disheartened or discouraged by any one study of any one drug. In fact, recognize that we are the beginning of this journey, and it is in fact a marathon. And while any one of these drugs will be successful or not in any one trial, I would say to you, do not get discouraged or disheartened. We are at the cutting edge of this science. No question but that this is the future of medicine. And hang in there with us, hang in with the studies, hang in with your doctors taking care of you, hang in with this wonderful group, and recognize that in the long term we will win this battle. Thank you very much.
Our next speaker is Xavier Loria, who's Medical Affairs Director from Santhera Pharmaceutical, who is going to speak on Santhera's real-world experience with Idebenone in LHON, update on programs.
Good morning or good afternoon already, I lost count. Yes. I'm very honored and very pleased, and I'm very thankful to be here. I think it's a great audience and it's a great event, so I'm very happy to participate in it. I subscribe what Dr. Katz has said. I mean, we are all in for this, so we all put our little bit of information into the pot in order to cook something that can help everybody. What I'm going to tell you about today is, well, first of all, this is my disclosure. As you have heard, I've worked for Santhera. And one thing is I'm going to be talking about a product which is not available in the United States. It's not approved for use yet, but it's approved for use in Europe. And thanks to that approval, we've done further research. And what I'm going to present today is the latest results or findings that we've found analyzing the data. So I'm just going to take 3 steps. First, data on this expanded access program, as I said to you. Then for the post-authorization measures that the European authorities negotiated or imposed on us in terms of, yes, we will let you market your drug in Europe, but we want you to carry on collecting data, collecting evidence, because we still need to know more. So on that, those programs, I'm just going to give you a very brief update. And finally, I just want to highlight that there are external publications or publications by external experts which contribute to the knowledge and to the proper use of proper management of the disease, I would say. Right, just briefly to remind you about Idebenone, antioxidant. The problem is complex 1 in LHON. Electrons are there in a kind of a traffic jam. They cannot circulate to the rest of the respiratory chain. That causes problems, reactive oxygen species, and on the other hand, the energy that is needed might not be produced in enough quantities. That causes, again, a problem in the retinal ganglion cells. Eventually, they might lead into apoptosis, but Dr. Newman has told you about that. So just trying to explain in a graphical way what is this, what is shuttling electrons. This is more or less what I found. This is a model, actually. So you would have a lot of commuters stranded and, you know, not going where to go. So someone decides to invent this thing. I don't know whether it is really working or not in real life, but you put the commuters on top of this thing and you just go over the cars. At the end of the day, you reach your destination, which is what you want to do. So a year ago, I was here and I was telling you about LHON, which is one of the post-authorization measures, one of the studies that we are running. The United States centers are participating on it. And because of that, I just gave you a brief introduction to the disease, which I don't need to do that now, about the drug, which I don't need to do that now. But one of the things that I can say is Dr. Katz is still going through the crossing of the desert in some way. You are in the research stage. We've gone past that. We've already reached the oasis in some way. There is water after all this thing, and we'll wait for you there. I mean, it is worth the effort to walk this path, but it is not easy. Was not easy for us. We designed a clinical trial that, well, could have been foolproof in some way or fail-proof. Well, it didn't get enough good results in terms of statistical significance, but it gave us a lot of learning. So I think that we all learned from that. Then we moved to the next step. Any kind of minute piece of information that you can get that can justify, explain, or provide more data to explain the results, the findings, that is very helpful. That's what we did with the Expanded Access Program. And then, very important, natural history. We think we know, we don't know enough. So every little bit of information is crucial for us, for any other company, and for the patients and physicians. Well, finally, we reached that other end of the tunnel. We were authorized, but again, we were asked to do further research, which is what we are doing. Not easy again because, well, placebo is, well, not accepted in some countries where the drug might be available. The other thing is, you know, we're talking about a rare disease, so not easy. In that sense, we need to deal with whatever we come across. And one of the things that we came across was, number one, the LHON study, which I'll touch briefly later on, and number two, the Expanded Access Program. Expanded Access Program started back in 2011, and it has a reason. When the first results were published, there was an interest in using the Idebenone in patients with LHON. So we had a number of requests by physicians worldwide in several areas and several countries in order to treat the patients, which we accepted, which we obviously provided the drug. There was no precondition in some way, so it was real clinical practice. So each physician would decide, except for the dose that we recommended, 900 milligrams per day in 3 divided doses, you know, how frequently they would see the doctor, how long they would treat. That was decided by the physician, the treating physician. But after a few years, we decided to collect data, because this is actually what happens in real life. And it's very important data. And that data helped us to support the claim of the product in front of the European authorities. But also now, in the latest cutoff, the last year, about a year ago, we collected again data, updated to that point, and this is what I'm going to present. Briefly, just to mention a couple of things. The way we measure efficacy is by outcome. Here is clinically relevant recovery. So a minimum change in the improvement of the visual acuity that is considered clinically meaningful by many physicians. Whether you are on chart or off chart, I'll show you now. But the other thing we also want to look is those patients that are still better than 2200, I mean better than less than 1 logmar, to try to keep those eyes in that visual acuity level so that they don't deteriorate into the legal blindness. This is the way we measure it. On the red side, let's say you see the clinically relevant recovery. We account for patients that start treatment off chart and patients that start treatment on chart. So that if a patient starts treatment off chart, it's not enough to come on chart, but we ask at least to see one line in the ETDRS chart or the equivalent logmar. If the patient is on chart, at least 10 letters improvement, that's 2 lines in the ETDRS chart, or 0.2 logmar. That would be for recovering lost vision. Now, for maintaining the current vision, which is less than 1 logmar, then what we ask is to, when you start the treatment, you are less than 1 logmar better than 2200, and by the end of the treatment or by the last observation, whenever it happens, you're still in that level of vision. So we got 111 patients treated worldwide. So in terms of safety, we have a nice number. Everything is okay in that sense. But in terms of efficacy, we only got 87 because some patients were treated and were never followed up. We also wanted to look at the main mutations, and what I'm showing on this table here is just the main characteristics of these 87 patients, which compose what we call the efficacy population. I'm not going to go in detail, but the ones that understand the data will see that this is very much in line with what we have in the real world for LHON patients. In terms of treatment duration, I just want to highlight that we have patients that were on treatment very, very short time, less than 3 months, but there are patients that have been on treatment for nearly 5 years or 5 years already. Now, then we ask the questions, in terms of preventing legal blindness, in terms of those patients that are still seeing better than 2200, can we prevent that? We saw some signs on the RODOS, the clinical trial that I've just briefly mentioned earlier on, but hold on a second, low numbers, 6 patients on the treatment side, that doesn't take us anywhere, no patients, 2 patients in placebo. But it's interesting to see that in the real world, we observe that kind of preventive of deterioration. So 50% of the patients that started with that level of visual acuity were prevented from crossing the threshold of legal blindness over this period of treatment. Now, the other question that we asked when we designed the analysis of this data was, can we recover visual acuity? So can we have clinically relevant recoveries? This is what I'm showing quickly here. And we see on the left of the screen the total of all patients. The average is 47% of the patients that were treated had a recovery of the visual acuity at that threshold of 2 lines when you are on chart, or they went from off chart, on chart, and able to read 1 line. So that's overall, and then we have the split by mutations, we see that effect is observed regardless of the mutation. Obviously, 1444 seems to be a better responder, but that doesn't mean that the other ones didn't respond. So, in terms of safety, as I said to you, nothing new. Whatever was shown in the RODOS clinical trial is still valid. We have basically nasopharyngitis, cough, mild to moderate diarrhea, but all of them mild to moderate, and no new safety concerns. Now, I could end here and say thank you very much, etc., but that would be a missed opportunity, because one of the important things that we realize is that when you have this bulk of data, you can still ask some more questions. You interrogate the data, and then you have interesting things. For example, we were told by some physicians that some patients seem to deteriorate at the beginning. So they seem to go down despite of therapy. So we wanted to look at the data and ask, is this true? Is this real? Well, on the screen you will see the three colors. If you can see them, it's the green would be the patients that are less than 1 logmar, so above the threshold of legal blindness. The yellow would be patients that are on chart but worse than 20 to 100, and in red you would have the eyes that are off chart. So on the red, if you follow the red lines, what you can see is that because these are individual eyes, and we know that these eyes, we only took the eyes that responded, so we have certainty that they had the CRR. We see that some eyes that later on responded, after starting the therapy, still went down into a kind of a nadir. So they still deteriorated. That is important, whether it is confirmed or not, to bear in mind, because that might induce some patients, some physicians to maybe overreact, to stop the treatment too soon. So the reading of this data would be, don't overreact, just let the treatment do, stay for a while, and then evaluate after a minimum time of treatment. The other thing is, can all eyes in some way, whatever they are in the visual acuity, respond? Without making any claim, what we can see here is the eyes that are on the red dots, which are showing eyes that are off chart, they can still, as we can see, they can still respond, and they can still respond in some cases to better than 20 to 100 logmar. So the reading for this would be, yeah, I mean, basically any eye can respond, so don't limit the application. Another very important question that we receive often is, how long should I treat before I see any effects? And what we've seen here is that some patients respond pretty quickly, 3-6 months, but other patients take longer. And here on the graph we show the global, but also the different mutations. And what I want to just draw your attention is that Rothers, the clinical trial, which was in theory perfectly designed, etc., only lasted 6 months. If you look at this graph with this real-world data, you see that 6 months can only account in the 11778 mutation, probably for about 40-something% of the patients that finally responded. So definitely, maybe we can say that Rothers was too short, and if we had to repeat it or do it again, I would probably go for longer. Here we confirm that the 14484 seems to respond faster, but you see, some patients can still take nearly 2 years to respond. So it's a long time. But this is an observation. This, I'm just put it here for the physicians that are used to this type of graph, just to see that a patient might respond, but the second eye might take still some time to respond. And this is what we see here. When the last patient has responded on the top line, we still see a few eyes responding after that. That's good to bear in mind. Now, the other question is, because we have physicians that treated the patients for a very, very long time, so we have long-term data. The question now is, if I keep on treating, do I reach a kind of a plateau in which there is no further improvement, or in fact, is there a further improvement at some point, or once you reach CRR, that's it? Well, what we see here is by mutation and by the total group, on the left you have, at the moment that the patient reaches the CRR, so we confirm that there is a CRR, we see the logmar indicating what is the magnitude of the improvement at that point. And then next to that we have the last visit, which gives us the magnitude of the improvement at the last visit whenever it happened, be 3 months later, be 5 years later. And what we see is that all across the mutations, there seems to be that an effect in which if you maintain the therapy, you can still reach further improvement. At some point, it's going to plateau. That's going to happen. But this is an important observation. Now, and just coming to a close, one of the things that we observed, trying to find signs on why some patients respond, why some patients don't respond, obviously without being able to make any claims here, we found that patients that didn't respond, maybe they were stopped too early. When you look at the graph, you see that at 6 months, the red line, which is the patients that didn't respond, were still there. But after 6 months, there is a huge and quick attrition of patients, which probably were stopped because of the lack of response. At that point, we were not sure about this 6 months, or that treatment needed to be longer. Anyway, coming to an end, the LHON, I wanted to tell you, it's still on. We are on course. We are 50% of the enrollment done. We are on the right path, and we expect to finish as planned, and that is going to provide very, very, very valuable information, because what I've shown you now, we will be able to confirm whether that is true or not in a very well controlled way. Anyway, for in terms of inclusion, exclusion, you can meet me outside offline, and we can talk about the criteria. But I would say that obviously the physicians that are participating in the LHON, those are the best source of information about this. Now, just to conclude, I would like to highlight this international consensus. It's very interesting, very important for the disease, because it's a way of drawing attention to the need of doing things in an organized, in a well-controlled way. There are going to be new clinical trials, there are going to be new products hopefully being successful, and that is going to require that we know what to do, when to do, how to do it at every single moment. And with that, I'm not going to put the conclusions, etc., etc. Thank you very much, and I'll take any questions later.
Our next speaker is Jim Carr, who is the Chief Clinical Development Officer of Stealth Biotherapeutics, and he will be speaking on LHON development update.
Hi, again, my name is Jim Carr. I lead the clinical development efforts at Stealth Biotherapeutics, and I'm delighted to be here today to provide a brief update on the progress of our LHON program. Our CEO gave a talk earlier today, so I'll just provide a brief overview of the company. We're a small biotech company based in Boston, just outside of Boston. Our focus is rare mitochondrial diseases, diseases associated with aging, as well as neurodegenerative diseases. We are a small company, but we have very diverse efforts really across the world. Dr. Enns actually provided a great overview about this, but when studying rare disease populations, there are many challenges which we have to confront, but so do our physicians, scientists, and in particular patients. There's a small number of patients to choose from, so I think everyone's aware of the epidemiology of LHON, so there just aren't that many patients. As a result of that, it's a burden for patients that have to travel across the US, for example. We had one clinical site for this trial in Southern California, so that puts a burden on patients and caregivers to get patients, really, if they're living in the eastern part of the US, to get to California, for example. Natural history is typically not well understood. Underdiagnosis is common, and phenotypic diversity within a disorder is, as you know, very common within mitochondrial diseases in general. The solutions are to build relationships with patients and patient advocacy groups, which is obviously why we're here. Patient registries help a lot to understand natural history, provides a mechanism to support the diagnosis, and then regulatory initiatives. So James Valentine gave a very nice overview of patient-focused drug development meetings, which really condition FDA to understand the diseases that we're hoping to get drugs approved for, drugs and devices. Protocols and clinical trial design are also complex in the setting. The challenges are there are a lack of well-defined and validated clinically meaningful endpoints. My background is that I've been doing really cardiovascular drug development for decades. I can do that in my sleep. There's really a blueprint for how to do that. One could argue that in rare diseases, we're kind of like sometimes building the airplane as we're flying it. So we launch it and we continue to optimize as we continue to evolve our programs. Again, there are small populations. We have to harmonize global trials. There's high subject burden, as we talked about. The solutions are increased engagement, collaboration with scientific advisors, clinical experts, understand, develop clinical endpoints specific for the patient population. So sometimes we have to build around endpoints. Explore unique trial designs and sometimes bring the trial to the patient. Now, a brief update about our program in labors. So, again, this was talked about previously, but our drug targets inner mitochondrial membrane and localizes the cardiolipin. We've shown in different disease models that we can essentially fix mitochondria, at least morphologically. It looks like we can help normalize the appearance of mitochondria. We believe going along with that, it leads to an improvement in mitochondrial function. We've demonstrated evidence of efficacy in multiple preclinical models and clinical trials. Now, this is the design of ReSight, the LHON clinical study design. This was a 12-patient trial. There were 3 different randomization schemes. So elimipritide 1% drops were delivered BID to both eyes in 4 subjects. 4 of the subjects received active treatment in the right eye, and the other eye got sham or placebo. And in the other group of patients, it was the left eye. So 4 patients got drops in the left eye while the opposite eye got placebo. The endpoints were safety and a variety of secondary endpoints. We are literally still analyzing the data. This trial just concluded recently, so I don't have data to share with you. I have some impressions about the data which I can share, but we will have, I wish I had as much data it was shared with you with GenSight, and we will have that very shortly. We're not there yet. The study population, again, there were 12 patients in this trial. The diagnosis of LHON was on the basis of clinical and ophthalmologic functional and anatomic test findings, satisfactory documentation of the DNA mutation, specifically 11778. It was confined to just that mutation. Loss of vision in both eyes of at least greater than a year and less than 10 years at time of screening. The investigational product was double masked, as I've talked about. It was elimipritide topical ophthalmic drops, 1%, delivered BID to the treatment eye. And as I talked about, there were 4 subjects that got drops in both eyes. 4 subjects, left eye, opposite eye, placebo, and again, 4 subjects, right eye, opposite eye, placebo. The vehicle solution was just matching in appearance, but it was a vehicle, a non-active vehicle. The patients, upon completion of the randomized or masked part of the trial, went into the open label extension, and that is still ongoing. These are the demographics, very similar to the demographics you've seen in previous presentations. The age was about 34 years. 10 of the 12 subjects were male. Time since genetically confirmed diagnosis, an average of 3.8 years. All subjects were randomized at least 2 years after the initial diagnosis. The BCVA at baseline is about 15 letters. This is more about some of the baseline characteristics. You can see that obviously very impaired vision at baseline in these subjects. And you can see the year of diagnosis. We didn't start this trial until 2016. So you can see that some of the patients were diagnosed in 2014. So most of these patients had been diagnosed several years prior to being randomized in this trial. As I said, we don't have detailed information about efficacy yet, but I can share with you impressions that we have. Again, we're still collecting and verifying data. I can say that signals of efficacy are apparent. There's preliminary evidence that long duration of dosing with drops will be a requirement. We could even tell by the mass data that it took several months before we could see movement on the endpoints that we were evaluating. There was a very low incidence of treatment emergent adverse events. So as James Valentine talked about earlier, for regulatory decisions, it's really a risk versus benefit. I can say unequivocally that it doesn't appear to be a very risky proposition to put eye drops, at least 1% eye drops in the eye in patients with LHON. In this trial, we're actually capturing videotapes from patients as they go into the open label aspect of the trial. This has been really interesting. What I can say is that already, I know we don't have all of the data yet, but I can tell that the functional vision or some of the benefits that are being described by patients, we probably don't have endpoints that are sensitive enough to capture that. What that may mean is that we need to develop a non-traditional customized endpoint which may capture some of the additional changes in vision that are meaningful to patients. So for example, I can provide an anecdote. There was a patient, and I've watched most of the videotapes. There was a patient that no longer trips over his dog while walking through the house. That's pretty meaningful. There was a subject that really enjoys eating and really appreciates the fact that he can pick out the things in the salad that he doesn't like. We can't capture endpoints like that. Right now we don't have endpoints that can capture that kind of specificity of what is meaningful to patients. So the videotape evidence is actually, again, really helpful for us to know how to develop endpoints that are important for the patients that have LHON. So our next steps, we are, as a company, remain committed to developing a therapy for LHON. We're in this for the long term. As was talked about, this is a marathon, it's not a sprint. We're aware of that, and we're up for the marathon. We've extended the duration of the open label extension on the basis of, again, our initial impression of the data. We're planning additional trials in both the US and China. We are considering evolving this to systemic dosing, which may address, among other things, non-ocular manifestations. We also know from animal studies we do get more drug to the back of the eye if we give the drug systemically. We know that we can get drug to the back of the eye with eye drops. We know we can get more to the back of the eye with a systemic delivery. And on the basis, we're hoping that the data are compelling enough to be actionable as they are. Again, we're still in the process of analyzing the data, but we're hoping the data are compelling enough to be actionable. At the very least, we'll be engaging with FDA to get agreement on our next steps for how we take this program forward. But as I talked about, one of the next steps, I think, is to develop an endpoint that is sensitive to the improvements in vision that are being characterized by the patients. So it takes a coalition of stakeholders. We're very aware of that. It's very humbling to present here amongst scientists and physicians that have devoted their career to studying some of the diseases that we're treating. So it's always very gratifying to collaborate with clinicians, scientists, but importantly, the patients that have to deal with the burden of the diseases we're talking about. We know there's nothing that would make us feel better as a company than to develop a treatment that can help patients hopefully obtain certainly a better quality of life and hopefully better vision. Thank you.